Now I will discuss about the process of protein synthesis. Decoding life, the process of protein synthesis. Translation is the cellular process where messenger RNA or mRNA is decoded to build a specific polypeptide chain. It occurs in ribosomes and involves three distinct stages. The first stage is initiation stage, second is elongation stage, and third is termination stage. This is intricate dance of molecular machinery that ensures that genetic code is accurately converted into functional proteins. The three stages of translation. There are three stages of translation. The first stage is initiation, the second is termination, and third is translation. The first stage of translation is initiation. Initiation is assembly of the translation in eukaryotes. Eukaryotic initiation factors or EIFs, that is eukaryotic initiation factor, help the 40S subunit scan for the IUG key factor. The key factors here is EIFs or initiation, eukaryotic initiation factor, 40S and 60S ribosomal subunits. Initiator is tRNA, start signal is AUG or methionine, and the energy source for it is GTP. It is for assembly and release of eukaryotic initiation factors. In prokaryotic site, there is Shine-Dalgarno sequence. Elongation, the sequential addition of amino acids. The ribosome moves 5' to 3' along mRNA, catalyzing peptide bond formation via ribosomal RNA or ribozyme. Key factor here is elongation factors or EFs, amino acid tRNA catalyst, rRNA or ribosomal RNA peptidase transferase, and its direction is 5' to 3' or N terminus to C terminus, and energy source here is GTP for tRNA binding and translocation. Now we will discuss about the process of termination. Termination, the completion of the polypeptide release factors ERFs recognize a stop codon, halting translation and triggering the release of the completed protein. The key factor here is eukaryotic release factors or ERFs, and stop signals here are UAA, UAG and UGA. These are the stop signals here, and energy source is GTP, that is required for factor release. The result is dissociation of ribosome subunits. Now the structure of ribosome. Ribosome has one large subunit and one small subunit. Large subunit has A site, P site and E site. A site contains amino acyl tRNA binding site. A site is amino acyl tRNA binding site. P site is peptidyl tRNA binding site, and E site is exit site. This is E site, P site and A site. And this is small ribosomal subunit, and there is mRNA binding site here. The ribosome machinery. A, P and E sites. The ribosome is divided into three functional sites that facilitate the APE flow of tRNA molecules during elongation. Essential roles and comparisons. The APE mnemonic: A for amino acyl and its incoming, P for peptidyl growing chain, E is for exit, empty tRNA leaves. Energy source is ATP versus GTP. ATP is used for tRNA activation while charging. GTP, that is another energy source that is used for gripping and going like translation. Ribosome sizes. Eukaryotes have 40S plus 60S, and that is even or 80S, and prokaryotes have prime, that is 30S plus 50S in the prokaryotes. Now we will discuss about the chaperone proteins. Chaperones are intracellular proteins that facilitate and maintain proper protein folding and preventing denaturing of proteins and misfolding. And for example, heat shock protein, it is also known as HSPs or heat shock proteins. for example like HSP60 or heat protein 60. These are expressed to protect protein denaturing. They protect proteins during stress, for example high temperature and low pH or hypoxia. Following folding sport, they ensure that nascent polypeptides reach their functional 3D conformation. So these are function of chaperone proteins.